Right. So this activity has to do with what we talked about the day before yesterday, which is cell transport. All right. We talked about 3 methods of cell transport. What were those, the big 3? Diffusion is one, probably the most important one. Okay. What was another one? Liam, what was another one? Starts with O. Osmosis. Okay. Okay. And Alyssa, what's the other one? 2 words. Uses energy. Someone else get it? Active transport. Okay. So these are the big 3. Okay. Diffusion, osmosis, and active transport. Now, the activity we're doing today is going to be focused on diffusion because we're just talking about transport within the cell. Okay. But quick review here. Okay. What are kind of the key points of each one of these? Well, for diffusion, all right, it's passive. Okay, so it requires no energy. It just happens, which also means the cell doesn't control it. Okay, it's movement from high concentration to low concentration. Okay, so in general, it's going to get things where they need to go. Okay, in general, it's going to get things where they need to go because they're needed where they're in low concentration, and where they're produced is where they're in high concentration. Okay, so it should work fairly well that way. What were the drawbacks of diffusion? Okay, remember, diffusion is kind of the shotgun approach. So I fire it and the pieces spread out as they travel further away. Okay, so what's the drawback? Can I tell the stuff exactly where to go with diffusion? Okay, so it's not targeted. And the further it has to go, the less of it is going to get there. Okay, it is not good over large distances, so not effective. Okay, and we said that wasn't really a big deal because cells are usually pretty what? Pretty small. Okay, since cells are pretty small, diffusion works pretty well over a distance of 100 micrometers. Okay, you get more than 100 micrometers, diffusion is going to become less and less effective. If you got a cell that's 600 micrometers across, it's going to die. Okay, it's just not going to work. All right, osmosis, okay, was a bit different because osmosis only moved one material. Okay, what was it, Sam? Who remembers? Water. Yeah, it moves water. Okay, so osmosis only moves water, and it moves it across the membrane. So it's not an internal, like from place to place within the cell kind of a transport. It's moving it across the membrane. It is also passive. Okay, and the reason the water moves is because of differences in salt concentrations. Okay, it moves to balanced salt. The salt can't move across the membrane, but the water can. So it'll move from the area where there's very little salt to the area where there is more salt. Okay, that could result in a cell like shriveling up or exploding, okay, depending on the situation. All right, how many people have heard of reverse osmosis? Okay, like if you buy like certain types of bottled water like Dasani and stuff like that, those are made by reverse osmosis. They essentially make osmosis work in reverse. So they put a pump on the water and they force it through a semi-permeable membrane, and the membrane catches all the salts and you get all this nice pure water on the other side. Okay, it doesn't naturally work that way. Osmosis would naturally move to balance the salts. This creates an imbalance by using energy and reversing it. We'll talk about that a different day. Okay, and then active transport uses energy, okay, and it pumps material across the membrane against the gradient. So active transport can make the concentration on one side of the membrane higher than on the other. The other two are always seeking a balance. Okay, so active transport is the opposite of that. All right, so those are our big three. Okay, so quick review of those big three. Today's activity is going to be about diffusion. Okay, diffusion and how the size of a cell can affect how well that process moves material. Okay, anybody need that one anymore? All right, so surface area to volume ratio. Okay, this is what I mean by surface area to volume ratio. The outside of a cell, in other words, its membrane, is its surface area. Okay, the surface area of a cell, the cell membrane, is responsible for essentially acquiring all the food and other necessary materials. Okay, because it's what allows them to come into the cell. Once they're in the cell, they have to be transported through the cell's internal volume by what process? Diffusion. Okay. Now, the bigger a cell gets, the bigger its surface area gets, obviously. Okay. And the same is true for its volume. But surface area is a two-dimensional measurement. Volume is a three-dimensional measurement, which means if I increase the size of something, its volume increases way faster than its surface area does. Okay? So a cell can still acquire more nutrients through a larger cell membrane. Its problem is that as it gets bigger, its ability to move those acquired materials is reduced because now its internal volume is so large that it takes forever for stuff to get from the membrane where it's acquired to wherever in the cell it's needed. Everyone kind of follow me on that? Okay. Calgary has exactly the same problem and right now is working to rectify that. The problem with Calgary is there are very few roads that go from one side of the city to the other, okay, uninterrupted. All right, they're building the ring road right now. Okay, obviously before they had Stony Trail, all we had to get through the city was Deerfoot. You guys probably don't remember that, but it sucked. Okay, when the only route through the entire city of Calgary was Deerfoot Trail from one end to the other, you could sometimes spend 2 hours going from the south end of Calgary to the north end of Calgary. Okay, because if anything happens, it would just shut down, right? It'd be slowed to a crawl. During rush hour, it was terrible, okay? But the more ways you have to get through, the faster it can go. So what we're looking at here is, let's say that this small cell here represents like a small town like Okotoks, okay? It takes like worst traffic conditions, 15 minutes to get from one side of Okotoks to the other, okay? So Okotoks represents a small cell. Calgary represents a very large cell, all right? So when a place like Okotoks or a small cell acquires some piece of material, okay, it only has to diffuse, about this far. So here's the material, it's acquired here. Even if its destination is way over here, that path is quite short. And as a result, diffusion would effectively get the material there. If we're talking about having to get the material from here to here, that distance is much greater. And we're not talking about one piece here. We're talking about a whole bunch of that material. What diffusion does, because it's not targeted, okay, it's not like a delivery service. It doesn't say, here's the address. It says, all right, we're inside. Just throw it everywhere and hope some of it gets there. Okay, well, what are the chances if I just throw material everywhere that by the shotgun approach, some of it will get here? Pretty slim. Do I have a better chance of that happening here in a smaller cell? Okay, so that's what we're looking at. This is the issue with diffusion. Diffusion works fine if cells are small. If cells get too big, their internal volume becomes a problem because diffusion is slow and it's not targeted. Okay, it's slow and it's not targeted. And it results in the material becoming less and less concentrated as it goes along. Okay, if you recall, the day I taught you that, I showed you the picture of the dye being put into that swimming pool. Okay, and you could see that right where the dye was going in, it was very dark purple. but even a short distance away from that, that purple color had gotten much less concentrated. Okay. All right, so does that kind of make sense? All right, so the background information that we need is this paragraph right here. Okay, cells absorb nutrients and excrete wastes. So this is kind of going both ways. It's not just about acquiring stuff, it's also about getting rid of waste. Okay, if you've got a bunch of waste products inside the cell and it's taking them a long time to get out, they can poison the cell. Okay, we want them out of there fast, right? So they do that through their membrane, which is their surface area, but they have to transport that through their cytoplasm, which is their internal volume. Okay, as a cell size increases, the volume increases faster than the surface area, and transport times take much longer. Okay, also the now comparably smaller surface area. So even though the surface area has gotten bigger, its ratio compared to the volume is now smaller. Okay, does everyone kind of follow me there? Even though it got bigger, the volume got bigger a lot faster. So the ratio of surface area to volume is now bad. It's small. Okay, we want lots of surface area and very little volume. We want a big surface area to volume ratio. As soon as that ratio starts to go in favor of volume, the cell will die. Okay, is everyone with me on that? Okay, diffusion is too slow to supply or meet the needs of very large cells, and as a result, cells do not typically reach large sizes. That's when they divide. Size is usually a deciding factor on whether a cell divides or not. Okay? Once it starts struggling with getting diffusion to do the job, it will divide into two smaller cells. Okay, now in terms of calculating, if we're gonna simplify this in this activity, and we're gonna deal with cells that are cubes. Okay, so that we can calculate their surface areas and volumes easily. Okay, if I want to calculate the surface area of a cube, I go length times width times the number of sides, 6. Everybody okay with that? Okay. Or if I had something other than a cube, I would have to find the surface area of all the sides and add them together. Okay. If I want to calculate the volume of any shape, it's length times width times height. Okay, so here's the issue with volume gets bigger faster than surface area. If I have a small cube, okay, and I calculate its surface area, I go length times width times height, okay, or sorry, times 6. If I calculate its volume, I go length times width times height. Now I make that cube bigger. Its surface area only gets bigger with these two numbers because it still has the same number of sides. But its volume had all three numbers that I'm multiplying together get bigger. Okay, so as a result, volume increases faster than surface area because it's three-dimensional. Okay, everybody all right with that idea? Okay, so here is the activity that we are going to do in class. We're going to do part of it. We'll do the calculations together here, so that'll save you time. Okay, so the problem we're investigating is how does the surface area relate to volume in cells of different sizes? Does dividing a cell into smaller cells affect the surface area to volume ratio? Well, we just kind of talked about that, and the answer to that is definitely. If a cell divides, does it solve its surface area to volume ratio problem? Yes, okay, it has just cut its volume in half, okay, and has not as much reduced its surface area, okay. All right, so for our hypothesis then, okay, our if part, okay, needs to talk about what happens to a cell, or sorry, how a cell transports material, okay, within its volume. What process does it use? Diffusion. Okay, so the if part here needs to talk about, okay, how a cell transports material, and I kind of just gave that away. Okay, so how it transports material internally. Okay, we talked about that already. The and part of this is we're going to mathematically compare cells of different shapes and sizes. Right, that's what this lab is going to look like. It's not really a lab, it's an activity. We're going to mathematically compare cells of different sizes and shapes. So we could say, and cells of different sizes and shapes are mathematically compared, or something like that. Okay. Our then part, okay, needs to talk about what we're going to see. Okay. Will cells that are bigger have poor surface area to volume ratios or not? Okay. and what is that going to mean for them? Okay, so okay, so will large cells, sorry, have poor surface area to volume ratios, and what does that mean for diffusion? That's what the then part of your hypothesis needs to talk about. Okay, and we've already talked about that. Okay, that's what we talked about here at the very beginning of class. All right, so we're going to start out with these 3 cubes. Okay, the first cube, micro, represents a normal-sized regular cell. Okay, we're going to say that it's 1 micrometer by 1, well, that's kind of small, but we'll just say it. 1 micrometer by 1 micrometer by 1 micrometer. Okay, everybody all right with that? Okay, so it's going to be 1 by 1 by 1. Macro is going to represent an abnormally large single cell. And it's going to be 10 by 10 by 10. Okay, so micro is going to be 1 by 1 by 1. Okay, this is going to be 10 by 10 by 10. Okay, and then multi-micro is going to represent what kind of an organism? A multicellular organism. Okay, it's made up of a whole bunch of normal-sized small cells. Okay, and it's going to have the same. It's going to be the same size as this abnormally large single cell. Okay, everybody with me on that? Okay, so what we're going to do right now is fill in the chart that you have there for surface area and volume ratios. Okay, and we'll do all the calculations together. Okay, I'm actually going to be in this way giving you 5 marks because there's 5 marks for having that chart right. So all you really have to do is write down the numbers here. Okay, all right, so we're going to calculate. So you have something, but it's much neater than this. Okay, looks like this, but it's much neater than this. All right, so for micro, I want to calculate the surface area of micro. It's 1 by 1 by 1, and it's a cube. So I'm going to go 1 times 1 times what? Times 6 to get the surface area, right? 1 times 1 will give me the surface area of one side, which will be 1, okay? And then I multiply that by 6. So the surface area of micro is going to be 6 micrometers square. Okay, there'll be 6 square micrometers of surface area. What's the volume? 1, right? Length times width times height. The volume of this tiny little cell is 1 cubic micrometer. Okay, so my surface area to volume ratio is 6 to 1. Is that good? Yes, that's large surface area, small volume. That's good. Okay, so the surface area to volume ratio of our first normal sized cell is 6 to 1. Okay, so those are good numbers. Now, I'm going to do the same thing for for macro. Okay, the abnormally large single-celled organism. So its surface area will be 10 times 10 times 6. All right, so 10 times 10 is 100, times 6 is 600. All right, so the surface area definitely got bigger. Oh, not cubed, squared. Okay, so its surface area got 100 times bigger. But what's its volume? 10 times 10 is 100, times 10 is 1,000. Okay, what just happened to my surface area to volume ratio? It just went from really good, ideal to pretty crappy, okay? Because now my surface area to volume ratio is 0.6 to 1. Now surface area is smaller than volume. Is that a good situation? No, That is far from ideal. Now there's a huge volume to transport materials through and comparatively small surface area to acquire and get rid of them. Okay, the tricky one is multi-micro. Multi-micro is the same size as macro, but it's made up of a bunch of tiny single cells. Okay. Each one of those cells has 6 sides. So what did we just do to the surface area? Well, we made it bigger. Yeah, we made it a lot bigger. Being made up of a bunch of single cells means every single one of those cells has a surface area all around it, okay? Whereas when I'm this big and I'm only one cell, I only have the very outside. This has the outside sides of every single cell, okay? So the surface area of this is actually 6,000. Because every single one of those little micros has a surface area of 6, and there's 1,000 of them in that cube. Okay, because it's 10 cubes by 10 cubes by 10 cubes. Okay, so there's 6,000 micrometers square worth of surface area in here. Okay, now it's the same size as macro, so its volume is still, it's the same, it's still 1,000. Okay, its internal volume is still 1,000. It's the same size. It's just made up of a bunch of smaller cells. All right, what just happened to my surface area to volume ratio? Went back to 6:1. This is why it's good to be multicellular. Okay, you can get big, but not have to worry about the surface area to volume ratio that is faced by really big single cells. Okay? You overcome that because you're made up of a bunch of small cells that have good surface area to volume ratios. Okay? Everybody with me there? All right, so we see that in general then, as a single cell gets bigger, its surface area to volume ratio gets worse. Okay? If we break that large cell into a bunch of small cells, then we fix the problem. Right? That's what that math is kind of supposed to tell us. Okay? All right, so there's 5 marks if you got all those numbers written down. Okay? Does everyone understand how we came about getting those numbers? All right, so what you guys have to do is this analysis and interpretation. Okay, so I'm just going to kind of quickly explain some of these questions to give you some tips on how to complete them. All right, so for question number 1, it's saying when the cube dimensions increase, they say centimeters here, but we did micrometers, from 1 to 10, that is, when I take the micro and turn it into macro, okay, the increase is a factor of 10. Each side is 10 times bigger, okay? But how many times bigger did the surface area get? How many times bigger did the volume get? So I'm just looking for 2 numbers here, okay? We just go back to our chart and go, okay, when I went from micro to macro, the surface area got how many times bigger, okay? When I went from micro to macro, the volume got how many times bigger, right? That's all we're looking for there. How many times bigger is it? Okay. For question number 2, it says the 2 big cubes, so macro and multi-micro, they are the same size. Okay, they say their size and volume have a factor of 1. That means they're the same size. Okay, how many times bigger is the surface area of multi-micro than macro? So again, you go back to the chart here and you go, okay, so multi-micro has this big, macro is this big. How many times bigger is that? Okay, that's what you're answering there. Okay, for question number 3, for which 2 cubes does the ratio between volume and surface area change when their size increases? Okay, so this is a bit tricky, this one. If I make micro bigger, it becomes macro. Does its surface area to volume ratio change? Okay, if I make macro even bigger, does its surface area to volume ratio change? Yeah, anytime I change one of these, it gets bigger, okay? But if I make a multicellular organism bigger, what does it do to fill in the extra space? It makes more cells. Are the cells all still the same size? So does its surface area to volume ratio change then? Okay, that's kind of what you need to put in for that answer. Okay, question number 4, okay, is asking you to calculate the surface area to volume ratios of these 3 shapes. So I've put the dimensions on here for you. Okay, but they're not in yours, but they're on here, so you'll have to copy them. Now, they're not cubes. Well, A is. It doesn't look like a cube, but it is. Okay. B is obviously not. It's 10 by 5 by 20. Okay. And C is obviously not. Okay. It's 1 millimeter by 90 by 10. Okay. So when you're calculating the surface area here, you can't just go length times width times 6. Okay. What you're going to have to do for the surface areas of these shapes, well, except for A, okay, is you'll have to find the surface area of like the top and multiply it by 2, because the bottom will be the same. Calculate the surface area of this side, multiply it by 2, because that side will be the same. And then the front and the back, same thing. Then add them all together. Everybody follow me there? And you'll have to do that for shape C as well. The purpose of question number 4 is to see if these cells' shapes can affect their surface area to volume ratio, because they actually all have very similar volumes. They're all very close. Okay, in terms of volume. They're not exact, but they're so close that it really doesn't make any difference. So they effectively all have the same volume, but does changing the shape of a cell help its surface area to volume ratio? That's what that question is trying to find out. So you have to calculate their surface area to volume ratios and then tell me, does shape affect the surface area to volume ratio? Give me a little explanation. That's what you're doing for question 4. Okay. Question number 5 then says, okay, so you just did this theoretical exercise with cells of different shapes. Here are cells within your body that are different shapes. Okay, how does their shape relate to what they do? All right, so we've got a liver cell. Okay, the liver, the cells are all essentially like A. Okay, they're small, they're fairly regularly shaped, but there's lots and lots of what in the liver? Okay, sorry. Yeah, yes, there's lots of smooth endoplasmic reticulum, but there's more blood in your liver than basically anywhere else. There's so many capillaries and stuff feeding the liver, okay, that its shape might not matter all that much because they're going to get fed on all sides effectively with nutrients. Okay, if you're a skin cell, which is this one, okay, do you get nutrients on all sides? Okay, so maybe you need to change your shape a little bit so you're more like B. Okay, you can talk about how that might help it, okay, doing its job. And then you got a nerve cell. You have some nerve cells that run from the base of your brain to the small of your back. One cell, okay? One cell that can be almost a meter long. But how thin is it? Very. It's more like C, because in order to feed these cells, blood vessels run along the spine. Okay? It's not trying to transport something from that end to that end. It'll transport it this way. Okay? So you got to kind of talk about that. How does shape affect, okay, what they do? And then number 6 is asking about the shape of a red blood cell. Okay, a red blood cell looks like a donut. Okay, so this is an electron microscope picture of it. So basically, what we're saying is, if a red blood cell was just a sphere, okay, would that have more or less surface area than a red blood cell that's shaped like this? Okay, because a red blood cell, when I look at it from the side, is essentially shaped like this. So which would be better? Which one would have more surface area? Okay, talk about that, because the role of a red blood cell is to carry oxygen on its surface area, right, on the membrane. That's what they do. So they're generally shaped like that. There's actually a sickness that some people have that's genetic called sickle cell anemia. And it makes actually their red blood cells look like a sickle, like a moon. Okay? How good would those be at carrying oxygen compared to the actual shape? Okay, that's kind of something to think about there. All right, then number 7 is asking, most materials get from place to place in the cell by diffusion. We talked about that. What effect would a low surface area to volume ratio have on intracellular transport? So, okay, we talked about that already. If we have a bad surface area to volume ratio, this is why that's going to be bad. Kind of explain, again, how does diffusion work? Why would that be bad? Okay, and then conclusion. Okay, so copy and paste your hypothesis and say whether what you found out here with all this math, okay, fits with what we said before. All right, so you've got the rest of class to do that. That's like 47 minutes or something like that. So that should be lots. Really, all you have to do is quickly finish your hypothesis, answer 7 questions, and write a conclusion. There's no sources of error. There's no variables. Okay, it's not a lab, right? It's an activity, right? So there's less to do here. So the expectation is that I have these submitted by 9:58. Okay. All right. If you have questions while you're working, just ask.